Hi, my name is Charlotte Coates, and I am here to present my project Looking Inside Plants to Understand the Effects of Climate Change, the Microgreenhouse Effect in Hollow Plants. My advisors are Dr. Peter Kevan and Dr. Jon Warland, and I am a master's student here at the School of Environmental Sciences at the University of Guelph. I have a question for you. What do all of these plants have in common? If you answered they are all grown in Ontario, you're partially correct. Or maybe you answered they are all enjoyed by people as food, ornamental, or recreational plants, and therefore they all have cultural and economic value. Well, you wouldn't be wrong if that was your answer either, but there's something else that they have in common. These crops are all plants that have hollow stems. Now that you're thinking about it, you may be able to name other hollow-stemmed plants that are all around us, like dandelions, asters, grasses, and horsetails. Given the significant number of hollow-stemmed plants, you would expect that scientists would have a good understanding of how hollow-stemmed plants are different than solid-stemmed plants. I'm here to tell you about an exciting new area of research that can be applied to hollow-stemmed plants to help farmers, growers, and conservationists understand plants' growth and development in new ways. This area of research is in the field of biophysics, and it is called endomicrometeorology. Endo refers to inside, micro means on a small scale, and meteorology, of course, means the study of climate. The areas that are well studied in plant micrometeorology already include atmospheric movements of wind and convection, the light conditions and radiation, temperature, humidity, and precipitation. And all of this information is used to apply to inform decisions about when to water, when to watch for disease and pests, and when to harvest the plants. Now, this is especially significant in controlled environments, especially here in Canada, where a lot of our food is produced in greenhouses. In field production, the technology is increasing to have better environmental monitoring as well, and these are used to inform decisions about when to plant seeds and watch for disease, etc. Given the new abilities in environmental monitoring, we must now ask, what micrometeorological conditions exist within plant parts? Now, the greenhouse warming of stems is an intuitive model of the greenhouse effect of warming in plants presented by Kevan et al. In 2018. The image on the right of the screen shows the pathways of heat transfer that occur within the stem. Now, these individual heat transfer processes are difficult to measure directly, so in my project I will address this by simplifying the model presented by Kevan et al. My project objectives are firstly to document the endomicrometeorology parameters of hollow stems. This includes the environmental data, plant morphology data, and plant tissue optics data. Next, I will create a simple heat transfer model using computational fluid dynamics software. Then I will aim to communicate my research and management impacts to farmers and greenhouse growers of hollow-stemmed plants. To achieve my three objectives, I have selected two hollow-stemmed model organisms to work with, and these are Gerbera jamesoni, shown on the left side of the screen, our field site grown at Van Geese Brothers Greenhouse in Grimsby, Ontario, and on the right side of the screen we have Cucurbita pepo, or pumpkins and squashes, which are grown at Strom's Squash Farm here in Guelph, Ontario. In my project, I will measure the temperatures inside hollow plants throughout their life cycles to create a true picture of how a plant's temperature changes on a variety of factors, including ambient air temperature, incoming light radiation, and the physical properties of plant tissues. For my first objective, to document these conditions, I am testing the hypothesis that hollow Gerbera stems and squash stems do not always take on the ambient air temperature. My methods to test this hypothesis are to collect temperature data within stems and to compare these using t-tests and ANOVA. To create my model, I will be testing my second hypothesis, which is that the temperature differences between the inside and the outside Gerbera and pumpkin stems are due to the combined factors of solar radiation and the biophysical properties of the stems. The method I am using for this objective is to input my measured parameters into CFD software to create the model using Fluent software. My third hypothesis is that hollow stems reach more extreme temperatures than the ambient environment, and the effects of these extreme temperatures for each organism can be identified with a proper application of biological knowledge. The methods to test this hypothesis are to test the model under different ambient air, light radiation, and stem characteristics and communicate these results to growers. To document the environmental parameters, the parameters that I am using are the irradiance of the sun and the orientation of the stem, which are measured by spectrometry and direct measurement. The ambient air temperature and the internal stem temperature are measured by wire thermocouples and omega data loggers. The wind speed will be measured by anemometer. The ambient humidity will be measured by handheld meters and the CR1000 data logger. Plant morphology parameters. The stem diameter and length will be measured by calipers and tape measure. The stem wall thickness will be measured by calipers. The hollow volume measured by direct measurement of water displacement. And the inside and outside wall texture will be measured qualitatively using the methods by Vogelmann 1993. The plant stem parameter of surface temperature is also important to measure, and this will be recorded using an infrared FLIR camera and FLIR software analysis using the methods by Byerley et al. 2020. You can see on this slide the infrared images of pumpkin peduncles on the left and the infrared image of Gerbera daisies on the right. The plant tissue optics parameters will be measured using spectrometry and the ocean optics software. The transmission of light, absorption, and reflection and emissivity of the stems will be measured using this method. You can see on the left, I have the spectrometer set up in the field with a plant showing the results on the right side of the screen, where you can see the spectrum of light which is passing through the stem walls. Plant tissue optics parameters are important to collect throughout the plant's growth development to compare this to the data collected from the temperature regimes of these plants. Next, I will combine the environmental, plant morphology, and plant optics data into a simple computational fluid dynamics model, also known as CFD model, which will be created in ANSYS Fluent software. The boundary and initial conditions of the model will be changed to reflect the different ambient air conditions. And I will assess the heat dispersion under different amounts of incoming radiation. The picture on the side of the slide shows the localized heating analysis model created in ANSYS Fluent of a simple tube which can be related to a hollow stem plant. And I will use this model to assess the heat dispersion based on the different stem characteristics that I have measured in the field. My third objective is to communicate the significance of my results. Now my model will determine how a hollow stem's internal temperature changes due to the ambient air around the stem, the solar radiation the plant is exposed to, and the plant tissue properties. So these results can be integrated into a grower's environmental management plan, which includes the irrigation, ventilation, pest management, and harvesting practices. This model will also provide the theoretical basis to understand how the effects of extreme weather conditions on hollow plants are different from solid stem plants based on this model of the CFD heat transfer. Thank you. If there are any questions or comments, please address me at coatesc@uofguelph.ca. Thank you so much. And now I have a list of references as well for this conclusion of the presentation. Thank you.